What if your cells had a metabolic backup system, not just to generate energy, but to defend against oxidative stress and build vital molecules? That's exactly what the pentose phosphate pathway, PPP, does. The PPP runs parallel to glycolysis in the cytoplasm and serves two main functions: generating NADPH, essential for reductive biosynthesis and antioxidant defense, and producing ribose 5-phosphate, crucial for nucleotide and nucleic acid synthesis. It begins with glucose 6-phosphate, which is oxidized by glucose 6-phosphate dehydrogenase to form 6-phosphoglucono-D-lactone, producing NADPH in the process. This step is tightly regulated and rate-limiting. The lactone is then hydrolyzed to 6-phosphogluconate and further oxidatively decarboxylated to ribulose 5-phosphate, yielding a second NADPH and releasing CO2. In the non-oxidative phase, ribulose 5-phosphate is converted into ribose 5-phosphate for nucleotide synthesis or transformed into glycolytic intermediates like glyceraldehyde 3-phosphate and fructose 6-phosphate through transketolase and transaldolase reactions. These enzymes shuffle carbon units between sugars, allowing flexibility depending on cellular demand. This pathway is especially vital in tissues with high biosynthetic activity, such as the liver, adrenal glands, and red blood cells. And where units where NADPH is essential to regenerate reduced glutathione and protect against oxidative damage. Understanding the PPP is key not only for grasping metabolism, but for appreciating how cells balance energy production, biosynthesis, and redox control. Subscribe and learn every day.